Good evening, everyone, and welcome to the Brian Crombie Radio Hour on Saga 960. I want to introduce you tonight to an interesting gentleman who's the CEO of his company and a really interesting therapy, a potential solution to cancer. I want to introduce you, if I could, to Roger White. He is the president and CEO of a Toronto-based company by the name of Theralase, and they've got a cancer solution, and they describe it as destroying cancer at the speed of light. Theralase Cancer Therapy in Search of Medicine's Holy Grail. Mr. White, welcome to the show, sir.
Hey, Brian, thanks for having me on.
So tell me, what is your holy grail, the speed of light cancer killer?
Well, the technology involves light-sensitive molecules that have an affinity to cancer cells. So they're attracted to cancer cells, they're absorbed by cancer cells, preferentially over healthy cells, and they're benign until light activated. So we instill these into the body. Currently, we're doing bladder cancer, so it's an intravesical installation. They locate into bladder cancer cells. We then light activate them. They destroy the cancer cells and leave healthy cells intact.
Sorry, what is it that's going into the actual cell? You're inserting something?
It's a study drug. So we have a small molecule, which is a powder, which we mix with water and it forms a solution, kind of an orangey red solution. This solution contains the drug, and the drug has the affinity for cancer cells.
And how do you make sure it goes to the right spot?
Well, that's kind of an interesting point of view. It's a mechanism of action. So cancer cells have a very high metabolic rate. So they have a very high need for iron. So there's a glycoprotein in everyone's body that transports molecular iron to every cell in your body, okay? If you look at the periodic table, there's a transitional 8 metal, which is iron. Right below that is ruthenium, right below that is osmium. The drugs that we produce are ruthenium and osmium based, our lead drug being ruthenium based. So what happens is the transferrin molecule, this molecule that's in everybody's body, can't tell the difference between molecular iron and our drug. So the glycoprotein transferrin joins with our drug and transports it preferentially to cancer cells. So cancer cells have what's called transferrin receptors. All cells have transferrin receptors, but cancer cells have anywhere from 20 to 100 times more than normal cells. So it's kind of a transport mechanism for our drug. And our drug is like a Trojan horse. So it goes to the transferrin receptor. The cell, the cancer cell, believes it's iron, so it freely accepts it into the membrane of the cell. It goes into the cytoplasm of the cell. It's benign until we light activate it. When we light activate it, it produces a very violent form of oxygen called singlet oxygen or reactive oxygen species, and it destroys the cancer cell from the inside out.
Fascinating. So our body's own molecules that take iron from wherever they are to fast-growing cells will attach to your drug, take it through the body straight to the cells that the cancer cells that are growing very quickly and are in need of iron. And I presume that this ruthenium, you said, doesn't supply the needed energy to the cancer drug that iron would, and so you've got it introduced into that cancer cell, and then somehow you activate it with light, and that light
correct.
Creates reactive oxygen species that then disintegrate the cancer cell?
Well, when you activate the ruthenium molecule, which is now inside the cancer cell, so you've got that transport mechanism, transferrin taking it to the cancer cell, activates, or it's inside the cancer cell. When light activates the ruthenium molecule, it produces singlet oxygen. So not oxygen is normally bound, O2, we all know that. But as singlet oxygen, it's very reactive. It wants to join. So it goes around like a wrecking ball, trying to destroy everything in its path, trying to bond to something. And it forces the cell through apoptosis, or natural cell death, or what's called immunogenic cell death. So it destroys the cancer cell from the inside out. It's a very unique way of destroying cancer.
That does sound very unique. Tell me, how does the light impact with the ruthenium to create the singlet oxygen?
With the ruthenium the actual small molecule is ruthenium centered, so it's got a ruthenium base, and it's got what's called a thiophene chain. So what happens is the thiophene chain acts like an antenna. So the light activates the antenna, sends it down the chain to the ruthenium molecule, and the ruthenium molecule produces singlet oxygen. So that's how it knocks out this oxygen. And because it's held inside the cancer cell, what we call spatially and temporally. So spatially, it's held inside the cancer cell, and it produces this singlet oxygen so quickly that the cell can't recover. So it destroys it from the inside out. So it goes through apoptosis. So the membrane would break, the internal mechanisms, the cytoplasm, the nuclei, the Golgi apparatus, anything that's inside the cell would then get thrown into the space, the interstitial space, and it would be absorbed by the immune system.
And then what, just naturally excreted from the body?
Yep, yeah, through the normal processes.
Really fascinating. It's almost like you're throwing a grenade right into the middle of a cancer cell.
That is the way it works. So because urothelial cells, we're talking about bladder cancer now, healthy urothelial cells don't have transferrin receptors, or a lot of them. They don't pick up the drug. And because healthy cells are able to, like the process for iron, molecular iron in a cell, takes about 20 minutes. So by the time the transferrin receptor gets to the cell, transports the iron inside the cell, and then leaves the cell again, it's about 20 minutes. If you're looking at cancer cells, that could be anywhere from 8 hours to 72 hours. So they are very, very slow at releasing the iron or releasing the ruthenium in this case. So all we really have to do is wait over 20 minutes, and then the cancer cell retains it. The healthy cell does not. We activate it with light, and we destroy it.
Now, how did you come up with this conclusion that light and ruthenium create singlet oxygen?
We're not the inventor of the technology. We've in-licensed the technology from a female chemistry professor. So over the life of the company, we've been working on this for about 20 years. We've in-licensed 3 different platforms of compounds. So the first platform we brought in from Virginia, Polytechnic University in Blacksburg, Virginia. So we brought in those compounds. They were really good compounds. They had a lot of energy, had some good destruction. They didn't have the best characteristics that we were looking for. We ended up winning, or Theralase ended up winning an award in popular mechanics for one of the top technologies in the US, I think in 2010. And we got contacted by another female chemistry professor, and she had 2 platforms of technology. So a ruthenium-based and an osmium-based. We ended up in licensing both of those platforms as well. And we started to develop those technologies, and they encompass thousands and thousands, if not millions, of different variations of these compounds. But through drug discovery, we found one very particular one we call TLD 1433, and we've now given it a brand name called Ruvidar. But that technology had a lot of promise, really, really good characteristics, things that we're looking for. You're looking for something that has a high affinity for cancer cells. It can bind with transferrin. It has a very high singlet oxygen yield. It's activatable in the visible red, or the visible wavelength, and also the near infrared wavelength. A number of different features. It's easily dissolvable into water. So these are all characteristics that we look for. And we started developing those with Princess Margaret Cancer Center out of University Health Network out of Toronto. We went through drug discovery. We went through in vitro, which is cell work, small animal in vivo work, which is small animal work. We went through toxicology. We went through phase 1 clinical study. And now we're in the middle of a phase 2 clinical study, about halfway through, which is a pivotal phase 2 clinical study. If we're successful, we'll end up getting marketing approval or commercial rights in Canada and the US.
Most people think that drug discovery takes a fair amount of time and a fair amount of money. Give me a sense, if you could, of how long this process has taken, will take, and how much are you investing in it?
The standards of thumb, or the rule of thumb for drug discovery from concept through to commercialization, is typically 15 years. and the cost to big pharma is about $1.4 billion US. We've been on this path for this particular set of compounds since 2011. So it's been about 12 years. It will take us another 2 or 3 years to finish the phase two, go through all the trials, get all the data in, do a data lock, get in front of Health Canada and the FDA. So the 15-year mark seems about right. As far as investment, we're not at the 1.4 billion US. We'll probably end up, I would say, putting about 100 million US Canadian into the project.
So about 1/10 the average, even 1/12 the average.
Yeah, yeah, much, much lower.
How
We
come
don't
you're
have
so much
the overhead.
cheaper?
We don't have the big overhead. I think with big pharma, what they've done over the years is they used to have huge research departments. So they used to have, 70,000 scientists or 50,000 scientists. So this huge overhead. And they used to develop their own drugs. But what they found is that, some of them work, some of them didn't work. I remember reading a story about a pharma company that put 900 million US into developing a drug and then ended up throwing it away because it didn't deliver on the characteristics they were looking for. So what big pharma does now is they have a lot of cash, they have big tills, and they look for winners. So they look for winners and losers, and they wait for the horse to cross the finish line, and then they just buy that horse. So why it's cheaper for us than them is because we're much more focused on a particular molecule and a particular disease versus big pharma, which may be looking at multiple diseases and maybe running multiple clinical studies. For example, the monoclonal antibodies are the big ones that they're using now, the atezos, the pembros, the opdivos, and these are molecules that they're using for so many different clinical studies that they'll run tens or hundreds of clinical studies at once. So that could add a lot to the cost.
Now, you've been with Theralase for quite a while. I think you and I bumped into each other about 15 years ago or something like that. And at the time, you were focused on low intensity light lasers.
That's
Tell me where that part of your business is.
Well, we started out in that business in 2004. That's how I got into the industry, where we were developing laser systems for eliminating pain, removing inflammation, accelerating tissue healing. So we're using red and near infrared light. We developed that into, a multi-million-dollar business, Canada and the US. We were selling internationally. And it's a good little core business. But I read an article, I think this was in probably late 1999, early 2000, about this female chemistry professor that was developing these compounds for cancer that were light activated. I became very interested in it. And I wrote to her, she sent me her research. I couldn't understand any of it. It was so complicated with chemical names, 15 inches long. And I became very interested in this technology, and I thought it had a lot of merit. So we continued running the therapeutic laser business, and we still do today. It's still part of our core business. But we put a lot of time and our effort in developing these anti-cancer technologies. We feel that that is a way forward. You're looking at subsets of patients and disease states that don't have any good outcome measures. Like there are developments happening, but the standard of care has traditionally been surgery, chemotherapy, radiation. These have a lot of side effects. They're very hard on a patient's body. They destroy a lot of healthy cells. So we became very interested in technology that would be able to destroy cancer cells, but leave healthy cells intact and be able to destroy the disease for the patient.
Your experience with the light laser, low intensity light laser, must have helped you with this cancer treatment and the light activation of the drug. Is that true?
It allowed us to pivot because knowing that technology and building laser systems, we're able to build a laser system. So the drugs are in license. So we in-license the drugs. We have them manufactured at GMP facilities or good manufacturing practices facilities in Canada and the US. The lasers are all developed by us. So all the laser systems, the fiber optics are all developed with our team of engineers.
You're a public company?
I am, or we are.
The ticker is TSAE. TSXV on the Venture Exchange?
It's TLT on TSXV.
Oh,
Ticker's
okay.
TLT. And
TLT.
we're OTC as well, QB. We're TLTFF.
Excellent. And you're at the 35 cents and about $70 million in market cap. Is that correct?
I think we're a little lower than that. I think we're about 26.5 cents Canadian and probably a 57 million market cap.
Oh, okay. Your website is a little bit out of date then. I apologize.
I probably is out of date. Yeah, I think the current trade, we're a little lower than that.
Okay. And obviously you can't talk a lot about your prospects, but obviously you're positive on this drug and the impact on your share price.
Well, I think that the technology has a lot of merit. Our latest clinical study or clinical study results that came out May 30th show some impressive results. So the FDA put out guidance in 2018 on a very specific subset of non-muscle invasive bladder cancer, which they call BCG unresponsive, non-muscle invasive bladder cancer carcinoma in situ. So the standard of care is a bacteria that was originally developed for tuberculosis called Bacillus calmette-guerin. So they used a watered down version of that, which they intravesically inject in your bladder, and it has a lot of results. So in non-muscle invasive bladder cancer, 75% of the patients respond, 25% don't. So they're considered BCG unresponsive. Problem with BCG is A, it's in short supply in the world, and B, the recurrence rate's about 50% at 12 months. So although 75% would respond positively, half of those patients recur again at 1 year.
It just comes
So
back.
Sorry?
It just comes back.
Comes back, it recurs. So it's so the FDA put out guidance in 2018 saying, well, what do we do about this population of BCG unresponsive NMIBC? And they said, let's make it tougher. We're only going to go to high-grade disease, which is called carcinoma in situ, not papillary disease, which would be on the inside of the bladder, but it's kind of like a red shag carpet on the inside of the bladder. So they made it very, very tough, very, very small subset. But they said these patients don't have a lot of options. They're going, if they fail BCG, they're going for radical cystectomy, which is a removal of the bladder, removal of various female and male genitals, removal of lymph nodes. It's a very intense surgery. So it has a high morbidity, high mortality rate. So they said, what do we do with this population? So they put this challenge out to industry. Theralase is one of the companies that addressed it. So we came up with this drug and this device in order to destroy it. So they were looking at, the international bladder community said, well, if you could get a response in half of those patients, you could give them a complete response at any point in time, and then you could keep that for half of that amount or 25% of the original population at 1 year, that would be a huge win. We would have something that would be widely adopted across the medical community internationally. So there's a drug called Vicinium, which was approved by the FDA, I think, 15 years ago. And it had a kind of a dismal response. At 12 months, I think it was about 7 to 10% complete response rate. So only 1 in 10 patients would be able to still have a complete response. The second drug that got approved was by Merck, their Keytruda drug. And it got approved in January 2020. And it had a 41% complete response rate at any point in time. But at 1 year, it was around 19%. So a little low, but they still got approval. The latest data that Theralase put out, we had a 66% complete response rate at any point in time with a 33% at 15 months. So dramatically higher than what's already been approved. So we think it bodes well for the future of the company. And as it bodes well for value of the organization, hopefully it bodes well for the shareholders and the stakeholders in the company as well.
And was this in your phase 1?
We're in the middle of a registration trial, phase 2, halfway through.
And so this results that you published were from your phase 2 trial?
That's correct. So
and how many people are enrolled in your phase 2?
We're looking to treat 100 to 125 patients. Okay. We've treated almost 60, 59 to date. So we're looking to wrap up that study end of this year, beginning of next year. With that, the last patient in, we'd have to wait 15 months to follow them up because that's the study design. So we'd be looking to have a complete data set, in about 15 or about first, second quarter, January, or first, second quarter of 2025. And then from there, we'd be looking at either a 6 or 10-month approval, depending on whether the company was able to achieve priority review with the FDA.
So
So I
you
would
would
say
only
end of 2025, beginning of 2026.
need a phase 2. You wouldn't need a phase 3.
No, because of the guidelines that the FDA put out, with this, you can do what's called you can compare your active arm to historical standards. So for this patient population, because they have no other options, it would be unethical, for example, to give them not the drug and not light activated. So the medical community and the FDA and Health Canada agrees that you could go across in a historical standard. So why in a phase 2, the FDA, again, in their guidelines, if you're running an intravesical installation, so you're putting the drug in through the urethra into the bladder, you can go and use a single arm study. You don't have to go to a phase 3. You don't have to go to a double arm study. It could be an open label study.
That's interesting. We're going to take a break for some messages and come back in just 2 minutes with Roger White talking about Theralase, the company, and his holy grail of a potential light-activated drug therapy to destroy cancer in bladders. Stay with us, everyone. We'll be back in 2 minutes. Welcome back, everyone, to the Brian Crombie Radio Hour on Saga 960. We're chatting tonight with Roger White. He is the president and CEO of Theralase. They've got an anti-cancer therapy that targets and destroys cancer cells, and they're looking right now at bladder. And it's really quite interesting because what they do is they inject into you, I guess through the urethra, you say, a drug that is a powder mixed with a liquid, and then it's injected into the body. It is ruthenium, which is like iron, and there's proteins, there's molecules in our body that carry iron, and will also carry ruthenium to cells that are in need of iron, cancer cells that are in need of a lot amount of iron. And so therefore, there's a natural mechanism that takes this drug from where it's injected into our body to those cancer cells. And then once it's there in those cancer cells, it goes into those cancer cells. And Roger and Theralase shine light on it. The light activates the drug. The drug turns into radical oxygen species, singular oxygen, and that destroys the cancer cells. So it's like putting a grenade into the middle of the cancer cells and blowing them up. Absolutely fascinating. He's gone through a phase 1. He's in the middle of a phase 2 clinical trial. He thinks he's going to get results by, I think you said 2025. And so therefore you might get approval thereafter. You're going to spend about $100 million, about 1/12 of the 1.2 that typical big pharma spend. And while you're at that 15-year sort of time frame, if you actually get approval in 2025 and not have to do a phase 3 in totality, particularly from today, it's a lot shorter than what a lot of drug candidates go through. So that's great. Now, tell me about your phase 2. You're about halfway through, maybe a little bit above halfway through your recruitment. Why is it taking so long to recruit all your patients? Is it a very rare cancer that difficult to find patients that will enter the trial?
It is. When you're dealing with if you look at Canada, we'll talk about Canada, there's about 13,300 new bladder patients every year. About 75% of them are non-muscle invasive bladder cancer. So 25% are muscle invasive. So the disease has gone into the detrusor muscle. Those patients are scheduled for radical cystectomies or having their bladder removed. Of that remainder, the 75% of the 13,000, there's only about 5% of those which are carcinoma in situ. So you're getting into smaller and smaller numbers. And when you look at that, those patients are spread all across Canada. So they're not in like one location or in Toronto or Vancouver or wherever they may be. So it's a very unique subset. So the reason that the trial takes a long time is in Canada and the US, it's a very unique patient population, or it's an orphan subset. of non-muscle invasive bladder cancer. Patients don't know about our study. There's also competitive studies. So there's other studies run by Big Pharma that are using other therapies, monoclonal antibodies, et cetera. So it's hard to enroll in this study. So it does take quite a bit of time. We started it in August 2019. We were going really, really well. We had enrolled, I think, about 12 patients up until March 2020. And then this wonderful thing came along called the global COVID-19 pandemic and shut everything down. It shut the clinical study sites down, the hospitals, everybody attention turned to this new virus. So that slowed down our process. Things started picking up again in late 2020, into middle of 2021. Things became a little bit more normal, I think, at the end of 2022 into 2023. So things have slowed down. So it's a very select patient population. And we had almost 3 years of a pandemic slow us down.
You've got about a 50 million market cap today, you say.
Approximately, yeah.
If you were approved, what would be your potential revenue?
Well, the market size, I don't know what our revenue would be, but the market we're looking at, the addressable market, is anywhere from about $1 billion US to around 11 billion US per year. So depending on the market, if you're looking at just the US, it's probably in the 1 to 1.5 billion range. If you're looking at internationally, it could be up into the 7 to the 11 billion. So there's a lot of data that's coming out saying that bladder cancer is highly linked with smoking. So if you look at the smoking and somebody has a cigarette or whatever their tobacco choice is, the body's very efficient at processing that. So the nicotine goes to the satiety center in the brain. It gives you that rush, which is great. But all the nasty chemicals, which are in that cigarette smoke, get filtered out through the kidney, drop down the ureter into the bladder. These carcinogenic materials sit very close to the bladder wall. So they, over time, can erode that bladder wall, destroy the urothelial layer, and eventually introduce these carcinogenic materials very close to your bladder cancer cell or bladder cells and make them bladder cancer cells. So what's happening, I think, in today's world is that there has been a lot of news I've seen in developing countries and a lot of other countries for people smoking. There's also been a high prevalence of young female smokers. So bladder cancer has traditionally been a male disease. So it's the 10th most common in the world, 6th in men, 17th in women. So traditionally it's been associated with men, older men. It's generally an older individual's disease, generally happening after the age of 60, but not always, in men. And I think with the prevalence of female smokers, young female smokers, as this goes on decade after decade, I think you're starting to see a higher prevalence of smoking among that population.
Do you take this treatment, this drug once or more than once?
This is a one and done theory. So the technology, it's not like an intravesical or an intravenous drug that you take every 2 weeks or 3 weeks. This is a in-office procedure, which is done in the operating room. So the patient, for example, let's say they had bladder cancer, unfortunately, God forbid, and they were coming in for this therapy. They would come in at 7 o'clock in the morning. They would have the drug instilled in their bodies, in their bladder at around 8 o'clock in the morning. They would go into OR. They would be put under general anesthetic. They would have their bladder flushed. Then they would have a metal cystoscope inserted, which is what the uro-oncologist uses to see inside the bladder. They would have the bladder filled with water. You would then insert a fiber optic assembly, which is going to be the light activation. So the drug has been instilled in their bladder from 8 o'clock to 9 o'clock. They now go in for the study device section. So from 9 o'clock to around 10:30, the drug is activated. The bladder's flushed. They are woken up out of general. They go into the recovery room and they get to go home in the afternoon. So it's a very unique process, which is a one-day event.
Right. And to get approved in Canada, you would need to, number one, get approval from Health Canada and then get on the provincial formularies to put you on their formularies in the United States, the FDA, and then what, you'd need to go to all the managed care and people to try to get reimbursement. Is that correct?
Well, the first thing I believe, and you're correct, is to get the regulatory approval, so the Health Canada and the FDA. After that, yes, have to get on the formulary, and you'd have to get on the insurance companies and the providers and look at it. You would have the approval, but now you face the battle of how much are they willing to pay for this, and
So
that's
how much do you think you would charge for it wouldn't be an issue in Canada if you got the provincial formulary approval, but how much would you charge in the United States, do you think?
It's hard to say, but you'd have to look at standard of care. So a lot of the monoclonal antibodies, currently they're running about 10 to 15,000 US per month. So if you're on that for a year or 2 years, you're running into the hundreds and hundreds of thousands of dollars. I think it's realistic to say that something like this, may cost a few $100,000 in order to deliver this. It depends. You'd have to put it in context with the population, what's currently being charged, the duration of the response, etc.
That's a hefty figure, but I guess if it takes care of cancer, it's worth it. This idea that a light activated drug could destroy cancer cells must have applicability beyond bladder.
It does. We are actually developing a intravenous or an IV installation of the drug. We've completed all the work, all the preclinical work, and we're just going into what's called toxicology analysis. So we have to look at the different levels of the drug and what are the safe levels for human use. Having completed that, we plan to start a phase 1 clinical study for glioblastoma multiforme, which is a very deadly form of brain cancer, and for non-small cell lung cancer.
Well, good luck. This sounds fantastic, and I really hope you're successful for benefit of all of us.
Absolutely, for all the patients. That's our primary focus, is for patient betterment and to deal with a deadly disease and to provide this technology Canadian-made.
If people are interested in your study and think that they could enroll, what's the process? Is there a website they can go to enroll in the study?
There is, I think the easiest way to go is if you go to our website, www.theralase.com. That's a good resource. From there, there's also a clinical trials registry called www.clinicaltrials.org. And if you look up Theralase, you'll be able to see all the clinical study sites which are enrolling and treating patients in our study. In Canada, I can tell you it's McGill University out of Montreal. It's University Health Network, or Princess Margaret, out of Toronto. There's Nova Scotia Health Authority out of Halifax, Nova Scotia. There's London, Ontario, so the main hospital in London. And there's also Vancouver Coastal Health, or the University of British Columbia in Vancouver. So there's 5 sites in Canada.
And what about your stock? If you're interested in the stock, where can one get investor information?
You can contact Theralase.com. You can contact our investor relations department. You can reach out to myself or the CFO or the Chief Scientific Officer. And absolutely. So we're actually in the middle of a raise at the moment. So if they're interested, they can certainly reach out to us.
How much are you raising right now?
Up to $5 million.
And have you got a prospectus with the OSC, or?
No, this is a non-brokered private placement.
Okay, excellent. Well, Roger White, it's a pleasure to meet you again and chat with you about your cancer treatment, and best of luck, because these are the kinds of treatments that we need to have approved and on the market and saving people's lives.
That's perfect. I appreciate your time, Brian. It was great.
That's our show for tonight, everybody. Thanks for joining us. I'm on Monday through Friday at 6 o'clock on 960 AM. You can stream me online at www.saga960am.ca. Good night, everybody.